Well, hello, welcome to everybody, good morning. We will introduce our first speaker, which is Dr. Wendy Gonzalez from Chile. She currently works in the Center of Bioinformatics, Simulation and Modeling at the University of Talca in Chile. Wendy does research in biophysics and molecular modeling of ion channel and their interaction with drugs. Her current project is structural insight into the mechanism of multi-channel blockers of atrial ion channels. Also, Dr. Gonzalez is co-researcher of the millennium nucleus of the ion channel-associated diseases. Welcome, Wendy. You can start when you're ready.
Can you hear me now?
Yeah.
Okay, and can you see the presentation now? I will present now. Wait a minute. Okay.
Yeah.
There we are. Okay. Let minimize this Zoom. That is okay. Okay. Now I will present my job about the molecular modeling of ion channel associated diseases. Good morning to all of you. And okay, here, okay. It's important to notice in a Latin American Congress that I was born in Cuba, and I started my bioinformatics research and formation here in the Center for Genetic Engineering and Biotechnology in Havana, Cuba, although I'm since 2004 in Chile. Well, I'll tell you about ion channels associated diseases, and these ion channels related to diseases are related because they have defects in their function, and the defects come from channelopathies, so mutations within the channels and the dysregulation of their expression. And how do we study ion channels? By means of microscopic or atomistic models using molecular mechanics. And usually what we have is proteins with about 7,000 atoms. And when we embed our ion channel on membrane and with water around and ions, we have systems of about 100,000 atoms. There we implement computational approach to study these channels, for example, molecular dynamic simulations, and to understand the interaction between the drugs and the channels. We implemented molecular docking, and I will focus in this third point, which is the pharmacophore design based in drugs, known drugs. We design pharmacophore, and with this pharmacophore, we look for compounds that share this pharmacophore, and what we do is to perform virtual screenings to find, for example, new drugs targeting these channels. I will focus in my talk in the pharmacophore concept, and I will go beyond, showing here 3 publications that we have in this topic since 2015. But I will mention briefly what we did here in this science paper, where we describe the pharmacophore of negatively charged activators of 3 different kinds of potassium channels. We are studying the drug ion channel interaction because this pharmacophore, particularly of the negatively charged activators and others that we have been described, may be exploited in rational drug design. We are, I think that since 2012, trying to find or to understand these drug interactions with ion channels. And we started this rational drug designs trying to find selective drug for a kind of potassium channels, the family known as K2P channels. But nowadays, we are going beyond and trying to understand how promiscuous drug works, because we noticed that several, for example, anti-arrhythmic drugs are promiscuous, such as carvedilol or amiodarone. And nowadays we are focusing in this topic, but it's important to notice that at the beginning we wanted to go and understand selective K2P drugs for K2P channels. Yeah, I must mention that these channels are targeted of very interesting drugs, and they are also evolved in this pandemia, because SARS-CoV-2 also have ion channels, such as envelope protein and ORF3A protein that we are currently studying, and also could be target of some therapies to avoid, this situation. And going to the science paper that I will mention, more or less what we did, you can see here 3 kinds of activators. At the beginning, or before this publication, these 3 activators were, or the scientists thought that they were only K2P activators. These 3 belong to HER activators, and these 2 BK activators. But in this paper, we realized that K2P activators can also activate HER channels and also BK channels. And why? Because all of them share this pharmacophore. So all of them have this negatively charged group and these hydrophobic and aromatic groups that are separated by certain distances and angles. Why are they activators? Because they have this negatively charged group pointing to the pore of the potassium channel. So potassium is a positive ion, and through an electrostatic interactions, this negatively charged group attracts the potassium ions, and these 2 groups are embedded within the protein. And this is the first report in the literature of polypharmacological drugs targeting ion channels. That's why it's so relevant. And I must point again that, for example, one of these drugs, let's say BL1249, can activate simultaneously the K2P channels, HER and BK. And this is really important for further analysis that I will show you. A pharmacophore, that's what I will show you here, is an ensemble of steric and electronic features that are necessary to ensure the optimal supramolecular interactions with a specific biological target, in this case, an ion channel, and to trigger its biological response. But going beyond, we want to understand this not in the terms of ligands. Nowadays, we are trying to understand this in the terms of the targets. So if one drug, for example, BL1249, can activate these 3 channels, then they could bind in a similar binding site. So in an ensemble of steric and electronic features in the target that can interact with the ligand. This is what we call a receptor-4 model. And I will show you 2 examples, a receptor-4 model for antiarrhythmics and a receptor-4 model for local anesthetic binding site in cardiac ion channels. Why cardiac ion channels? Because there are many drugs that are, as I told you, in the market that point to 3 channels that I'll show you here. Here you can see the atrial action potential on the left and the ventricular action potential on the right. And with red rectangles, they are the channels that contribute to the action potential only in the atria, and that are not expressed, for example, KV1.5 in the ventricular. So to target for example, atrial fibrillation disease, which is related, is the most common arrhythmia in the Occidental world, the drugs targets these channels, but not only, for example, these two, KV1.5 and TASK-1, that are expressed or has high currents only in the atria, but also binds to NaV1.5 channel, okay? These drugs are, for example, I told you before, carvedilol. You can see here the IC50 in the different channels, and amiodarone, or this derivative, dronedarone. They are in the market, amiodarone and carvedilol. For example, AV0118 is also an antiarrhythmic, but it does not or it did not go beyond the phase 3 because it has some metabolic problems, but it's verified as an antiarrhythmic drug. And this is the example that I will use, and we will study compounds similar to AV0118. So I will show you the topology of these three ion channels, but the message is that even differing in their sequence and in their structure, they should share these local similarities in the form of the binding site for multi-ion channel blockers. And for example, drugs such as AV0118 that inhibit all three channels should bind in a comparable pocket. What about the topology of these channels? For example, KV1.5, assembly as a tetramer, you can see here, per subunit, they have 6 transmembrane segments, and S5 and S6 form the pore. Here you see the pore from an extracellular view with the ion in the center, and here are the rest of the segments that form what is called the voltage sensor. NaV1.5 is a monomer, but each domain from 1 to 4 resemble one subunit of KV1.5. And you see that the functional channel is very similar. On the other way, TASK-1 is a dimer. Why? Because TASK-1 has 4 segments, transmembrane segments per subunit, and 2 pore domains. Then, to assembly and form a functional channel, the functional channel has 4 pore domains, so it's only necessary that 2 of these subunit assembly to form the pore. And if you turn and check from a side view, these pore, they are very similar, and they are mainly similar in this 3 zone. This is the extracellular zone, number 1. This is the selectivity filter, and in the 3rd zone, they are really similar, and I will explain this. This is the central cavity, and around the central cavity, these drugs bind. Okay. So let's go to the example of the antiarrhythmic binding sites in cardiac ion channels. And in 2015, we showed that this AV0118 binds to TASK-1. It's more effective on TASK-1. The IC50 is lower than in KV1.5. And within this 3rd zone, we described 4 layers that are similar in the composition in the amino acid compositions. Nowadays, Fernando Vergara, a bioinformatician, is doing his thesis in pandemia times, and he redefined these layers by means of MD simulations and using the crystal structure of TASK-1. And then we define again these layers as well as the layers in KV1.5 channel. We still are working with the homology model of KV1.5, but we define again this by means of MD simulations. With these numbers, we go to find common binding sites for drugs such as AV0118. AV0118 has a pharmacophore that I'm showing here, and all of these drugs that also I'm showing, they have this pattern, this pharmacophore composed by an aromatic ring and 2 hydrogen bond acceptor groups. Okay, and in 2015 we showed that all of them has the same behavior. All of them binds better in TASK-1. I will show you another example. For example, A1899 is 67.5 better in the block of TASK-1 than in KV1.5. And this is the pattern of all these drugs that has this pharmacophore. And what we are describing also now is that by calculation of the balance descriptor, the pocket, these layers in TASK-1 are more hydrophobic than in KV1.5. And this could be the reason of the increased potency of these drugs in TASK-1. So also Fernando is calculating these layers that we, by a structural alignment, this is not published yet, but we did a structural alignment at the beginning in a homology model of NaV1.5 channel, and we found that at least these 2 layers, the second and the third, are also in the central cavity of NaV1.5, and they belong to the batrachotoxin binding site. Fernando is nowadays calculating this binding site by means of molecular dynamics and using the crystal structure of NaV1.5 channel that was reported also this year. So it's important to go back to the pharmacophore, because we are not only looking for the binding site properties of these 3 channels, we are also looking for new drugs that contain this pharmacophore and can bind to these 3 channels to develop new antiarrhythmic drugs. And this is the case of this drug that we call F2 and F3, mainly I would say F3, that were developed by Barbara Arevalo in her PhD thesis. Barbara synthesized at that time these drugs, thinking in selective drug for K2P channels. And you can see here the IC50 of these 2 drugs in TASK-1 channels. So F3 has 100 nanomolar IC50 in TASK-1 channel. But we realized that this drug also has this pharmacophore. So this drug could be a promiscuous drug that we can use to target these 3 atrial ion channels. And we did it. Mauricio, during his PhD thesis, measured the block of this drug in these 3 channels. He used 10 micromolar block, and we observed for sure that the block is greater in TASK-1, but this drug also blocks KV1.5 and also blocks NaV1.5 channels. So we have a new promiscuous drug that we will use to evaluate against atrial fibrillation in cardiomyocyte models later. Okay. The binding site in NaV1.5 is not known, and Juliette Mazzola, that is starting her PhD thesis, will describe this binding site in NaV1.5. And we know more or less the residues, and I show it to you the residues of the binding site in TASK-1 in KV1.5, but by site-directed mutagenesis, she will try to find the binding site in NaV1.5 channel. But Fernando did some or has some advanced work on that, and he found a pattern within these 3 channels. Okay, a pattern that Juliet will evaluate by site-directed mutagenesis, and I will tell you later how Fernando found it by a program that we have or that we call GeomeFinder. And you see here, this is the pattern, and in red, we have TASK-1 binding site that is verified already by site-directed mutagenesis. So these 3 residues in red. In green, also these 3 residues are verified, right, by site-directed mutagenesis in KV1.5. And Juliet will try now this phenylalanine, this valine, and also this one in blue that are from the putative binding site of this kind of drugs that contain this pharmacophore in NaV1.5 channel. And it's important to notice that this phenylalanine is crucial in the binding of many, many drugs. Also this valine. Where is this pattern? Is in one site, is in one site of this central cavity, okay? And to show you more or less the procedure, I will tell you about the receptor 4 model of the local anesthetic binding site in cardiac ion channels. In this case, what we used were 3 drugs, ropivacaine, lidocaine, and bupivacaine, local anesthetic, that they share this aromatic ring, this intermediate linkage, and they differ in this tertiary amine site. And we have the binding site already described in NaV1.5, also in TASK-1. We published this last year. And we are now publishing the ropivacaine binding site in KV1.5 in the British Journal of Pharmacology. It will appear soon. And with the known binding sites, we go further. And this is the protocol that we are using also in the other example that I gave you with the channel structures. With the docking, we describe the protein ligand interaction using PLIP, and we characterize the pocket and we compare the pocket. How we compare the pockets? By GeomeFinder, that is a tool that we developed here in Chile. That is why the flag is here. And also PLIP was developed by one of our alumni in Dresden. So here is the PLIP analysis, and you see that the drug is here in orange, and in green there are the residues that are already described in each binding site as part of the binding site, described by site-directed mutagenesis. And when we compare the binding sites, they are hydrophobic, and the hydrophobic pattern you see by this green surface in the binding sites. It's also important to notice that here in the PLIP interactions, we observe a pi-pi interaction always between a phenylalanine and this aromatic ring of the local anesthetic, a pi-pi parallel or perpendicular interaction. This is a feature that we consider is really important for the binding of the local anesthetic. Well, when we go to GeomeFinder, we found this pattern that is like a tetrahedron, but this side of the tetrahedron is the one that is already described by site-directed mutagenesis, and each of these 3 points are reported, and in all of these 3 points in each channel, we have a phenylalanine in this point, a leucine here, and an isoleucine here. How does GeomeFinder work? So it compares the binding site between TASK-1 and KV1.5, NaV and KV1.5, and TASK and NaV1.5, so by pairs. And it provides a score of the binding site. If we have 100%, we will have the maximal contribution of the non-bonding interaction of distances, parameter, and amino acid features of the binding site. But what we are observing that the steric and electronic features are more conserved than geometrical features in the receptophores. So the non-bonding interactions are very conserved, the amino acids for sure, because they generate these non-bonding interactions, but not the distance and the perimeter. So the geometrical features are not as well conserved in the receptophore as in the pharmacophores. And that's what we are observing also in the first project that I showed to you, in the antiarrhythmic binding site in cardiac ion channel. In this one, that we also consider that this binding site is not in the central pore, in the central cavity, but it's allosteric. It's on one side of the cavity, in some channels in the fenestrations or in the side pockets and other structures that are near the central cavity, but on the side. And also we are finding this in the binding side of these negatively charged activators in potassium channels. So we are doing also a follow-up of the science paper, and we are also having these found. Finally, I want to thank my group, my big group. Some of them are here listening today, and Ariela will also speak for us later. The funding institution, I want to thank my team in Germany, my collaborators that are really important in this task, the organizers of this Women Congress, and thanks to you for hearing me today under these difficult conditions for all of us. And bueno, que la ciencia los acompañe. May science be with you. Thanks.
Thank you very much, Wendy. I can turn on my, there is my camera. Wendy, I have a question for you. Yes. You recently said that steric and electronic interaction are more conserved than the geometrical features. So how do you evaluate this by molecular dynamics?
Okay, by molecular dynamics, what we try is to find this pattern. So imagine that we have 100 nanoseconds, and we divide the molecular dynamics in one structure per nanosecond. And then in each of these 100 structures, in each of this channel, we find a pattern. And then we try to compare, the pattern within all the molecular dynamic simulation, and then we try to find the pattern. In fact, there are some, for example, the one that I showed to you here, I will show again the presentation. Wait, that you are now you will see yourself. I think no, or but if I do this, okay. For example, this pattern was found in one frame regarding to another, so it was the best fit within all the molecular dynamic simulation. So we compare it. It's a comparison 100 per 100 frames, and then we cross the information, and this is the best that we have with the best score, So we use this MD simulation to perform this analysis, as I'm telling. Mm-hmm.
Well, Wendy, thank you very much for your presentation, for being here today with us. And we have time for questions. I think
I have a question. I don't know if I put it in the right place.
I can
you can make it loud, Chris, and you can open your camera also.
Okay, thank you, Wendy, for that amazing work and nice presentation. My question is 100 nanoseconds enough to be representative of all the conformation a channel may have?
It's an excellent question. So, you know, what we try to do first is to equilibrate our MD simulation, and then we go to a production, and then we try to find, this pattern within this 100 nanosecond and to compare the frames. But I think that they are enough because the drugs are described binding in a certain state, you know, and then what we are describing with the MD simulation is this state. We are not pretending to go from one state to the other with the MD simulation because it will take longer than 100 nanoseconds. What we are describing is the dynamic in the bound state of the drug. We know that is, for example, in NaV1.5 channel in the inactivated state, in TASK-1 in the open state, in KV1.5 also in the open state. And then that's the state that we are describing by means of MD simulation.
Thank you. And what you said came to me another question. Do you have different models for different states of the receptors? I mean, crystallographic models to start with different states? I mean,
Yes.
I don't know if the state, but conformations.
Yes, I mean, we started with the crystal structure of TASK-1, the crystal structure of NaV1.5, and with a model still we do not have crystal structure for KV1.5. We have a crystal structure for KV1.2. So we did many years ago, not me, but Samsung Group did the homology model, and then we are using this model. So we started with this, but in this crystal structure, we do not have the crystallized drug. So we bind the drug by docking, but considering that we know the binding site. We always know the binding site by means of alanine scan mutagenesis.
Thank you, Wendy.
There is another question in the yes. Chat. I don't know if I answer.
Yes, please answer. I will read the question for you. This question is from Maria Serrano. She says, hello, nice talk. And she said, in your MD simulation, did you consider water shell?
Well, I don't know if she means water shell of the water around the drug. If it is, we are doing. So we place the drug, we, besides we dock the drug. That's the first that we do, right? To dock the drug in the binding site, and then we apply waters, you know, in the protocol, we apply waters, and even that's one point that we are doing very, very well to try to find the waters that are in the binding site, because if we are talking about hydrophobicity, this is really important also. And then, yes, we are considering the waters around the binding site all the time. I think that's what she means with water shell.
She says that that was the question. Thank you very much. Well, Wendy, thank you for your amazing presentation.